men and women in this region of the brain, they really look quite similar. Well, what really surprised me is when we compared the cortex to chimpanzees and gorillas, our closest evolutionary relatives. And there we see something quite similar as across human individuals. The cell types are really quite conserved. Big motivation for building these atlases is to ultimately understand disease.
Hi, Dr. Trygve Bakken. Welcome to Press Insider. My name is Shireen. And, just to start off, can you introduce yourself and what you do?
Yeah, so my name is Trygve Bakken. I'm an assistant investigator at the Allen Institute for Brain Science. And I co-lead efforts to describe cellular diversity in the mammalian brain, focused primarily on the human and non-human primate, what makes humans the human brain the way it is. So this initially took a focus in the mouse brain to build up a really comprehensive atlas of cellular diversity in the mouse. And what this package of papers that is in Science Now really describe is moving from the mouse to a much larger brain, the human brain, and non-human primate brain, and building up those technologies to really describe the molecular diversity of cell types in those brains. And that required a lot of new methods development and has enabled a description that is, you probably saw in one of the studies that we describe over 3,000 cell types in the human brain that are all distinct based on their gene expression profiles. That enabled us to then ask, how do those cell types vary across individuals? What makes us the way we are versus someone else? And how do we vary from other species, including our closest evolutionary relatives? A big motivation for building these atlases is to ultimately understand disease and to see, what is the cellular landscape of sort of a neurotypical brain, and then associated with various brain diseases, how do those cell types differ from this reference? The study that looked at cell types, those 3,000 cell types, that really focused on 3 donors. So in these studies, we were not powered to look at those differences between sexes, for example, across the whole brain. We were able to, in an accompanying study, to look at one particular region of the brain, the neocortex that's involved in higher cognition. And there we had brain tissue that was donated by 75 individuals, and we could look for sex differences. And in fact, we don't really see much. So men and women in this region of the brain, they really look quite similar. Well, what really surprised me is when we compared the cortex, the region that I mentioned before, to chimpanzees and gorillas, our closest evolutionary relatives. And there we see something quite similar as across human individuals. The cell types are really quite conserved. There's the same kind of general parts list in the chimpanzee brain. But there again, we see differences in expression. And the surprise was those changes in expression really are about the wiring of that circuit. So it's how these cells are connected to each other and how those connections may change over time, the plasticity of those neuronal connections. Yeah, so we're fortunate that the NIH is continuing to support this work, the National Institute of Health in the US. So that is coming through this Brain Initiative Cell Atlas Network. Of course, this work involved a consortium of many different labs, and this new work is continuing those efforts to build an even more comprehensive atlas of the brain, especially to locate all these cell types in the brain. So one thing there's only sort of a coarse pinning of these cell types into different regions, And we really need to know exactly where everything is in the brain because that helps us understand the circuit. The other piece that's coming that's new is to be able to link these cell types to actual functional regions using brain imaging, like functional MRI. And so I think creating that link between cell type and function is another area that's where there'll be a lot of work going forward.